name is Maureen Hayden. I am a graduate student in the marine biology program at Texas A&M University. This video is being recorded for Project POEM through the University of Arizona and is funded by the National Science Foundation. This video is for the Industry Mentor project number 3, which is the shadowing portion of the project. As a graduate student, my work varies widely from day to day, so what I decided to do is, since one shadowing session wouldn't capture or encompass everything that I do, I've put together this film that kind of takes you through a portion of my project from beginning to end. So just a little bit of background, I'm doing my dissertation on plastic pollution in Texas and on how ingesting microplastics affects marine wildlife. But right now, this video is going to take you from sample collection at the beach all the way to some preliminary data analysis and kind of visualization. So I'm excited, and let's get started.
The camera now pans to a beach on a sunny day.
It is Saturday, September 14th, and my undergrad research assistant and I are out doing field work today. I'm currently panning the shore of Sea Rim State Park, and we're doing this under collection permit R01-04. So right now we are at one of our sample sites, and the way we did this is there's a dune walk, and then we go to the right and we go to this first trash can here. This is where we usually start what's called a transect, where we take a special meter stick and we'll go 50 meters parallel to the shore, so like 2 parallel lanes of traffic, and we'll look for the high tide line. Today with us we have 2 what are called quadrats, so these are PVC rectangles that we use, and each of them measure 0.5 by 0.5 meters. We have 2 long traffic marker poles that are about 4 feet high, and they're fluorescent orange to mark the beginning and the end of the transect. And then we have some small traffic flags, about 10 of these, that we're going to place along the line every 5 meters. And then we have over here some sample tubes for collecting, and I'll show you how we label those in a little bit with a Sharpie. And then we have my field journal, and in this took note of the date, where we are, and what the purpose of the trip was. And then I'll make other notes like the weather or if we find anything particularly interesting. And as always, our little baggie with sunscreen and bug spray, because you gotta stay protected. So that is the materials that we have, and I will take another video when we have the transect all set up. So to measure out our parallel transect line, we have this is actually called a transect. So you'll notice it has a handle on one side, and then it has a wheel of measuring tape with inches on one side and meters on the other. So this side is in inches, and then the other side is in meters. And let's see here if we can get to 1 meter for you. Here we go. So here's 1 meter, and then you can flip it over and you see that it is in feet. So we're going to be using the meter side, and the way we're going to do this is we're going to lay this out to 5 meters and just walk this spool every 5 meters and then mark it with one of those small orange flags.
The camera looks out on the beach at the
so
transect.
Here we are. We have set up our what's called a transect line. This is technically called a belt transect because the entire thing is in the shape of a rectangle. So it's 50 meters long, and then we have 2 of those rectangular PVC boxes I told you about on either side, and each one is half of a meter, so that makes for a total area, a big rectangle of 50 by meters by 1 meter. And so what we'll do is we have these 2 transect boxes, and they are going down the center. And so what we'll do is Charlotte and I will each sample, my undergrad and I will each sample in the area of our rectangle, and then when we're done we will flip it over like playing hopscotch, for example, and then we will keep going down the transect line. So for each 5 meters, we're going to be doing 10 of these transects for each one for a total of 100 samples, right, on each side, so that's a total of 200. And then we're going to go all the way down to the end. So you'll see that we have it marked here by this big traffic pole, and then we will keep walking down, and every 5 meters or so you'll see one of these orange flags. This helps us keep our bearing and go in a straight line. So here's another flag, come up on 5 meters. And right now, if I were to describe the scenery to you, we have dunes with vegetation, about maybe 3 feet of dry sand, and then where we set up our transect line is at high tide. So what that means is it goes from being dry sand to slightly damp. So this is the point at the beach where the waves will come the closest to the dunes. And right now, we decided to come to the beach at low tide, where the waves will be the furthest away from the dunes. And Texas is one of those interesting places where we don't have a regular high and low tide. It's irregular. So the high and low tide doesn't come at the same time of every day. It comes differently. So we always want to try to time our samples around low tide, so that way our high tide line, or that point where the waves come closest to the vegetation, is easily visible. So again, kind of backtracking, if we keep going along here, we're just going to keep sampling along this transect every 5 meters until we get to the end, and we have marked the end of it by placing several flags in that one spot. So we're going to start sampling. So we're going to start collecting, and to do this we have a 50 milliliter Falcon centrifuge tube. It's just a plastic test tube. To give you an idea, it's about the length of the palm of my hand and about the width of my ring finger. And then I just have some small tweezers like you might find in your bathroom or your household. And what we're going to do is we're going to collect any piece of plastic that we find on the surface of the sand here, and we're going to put it in our test tube, and then we're going to take it back to the lab for processing.
Back at Texas A&M University in College Station, Maureen labels test tubes from their sampling trip with a Sharpie.
Okay. Here we have our setup to look at microplastics. So for this we have a dissecting microscope, which has an open platform stage. Mounted on that stage we have a plate with a black background for contrast, a ruler, and then we have our test tubes with our samples, a dish which we can put some DI water in, deionized water to rinse samples off. We have a light source, and then we also have our laptop. So on our top of our microscope, on one of our ocular lenses, the part that you look through, I have a camera pen called an iPivo point to view camera pen, and it's connected with a USB port to my laptop, and then I use something called Presenter software, and this allows me to see what's being projected on the microscope on my laptop. And the cool thing about this is, one, you don't have to strain your neck, and two, you can take this image and you can take pictures or video, you can live stream. It's pretty neat. So when we're looking at these microplastics, one thing that's really useful is we can put both the ruler and our sample in the frame. So for example, I have a piece of plastic on what's called the stage or the platform of the microscope right now, and then if we look through the image here, I can place it up on the black background for contrast against our ruler, and then there's a button here to take a picture, so I can do that, and I can save these for later and use a processing program to get a more accurate measurement of each of our samples. So for example, to show you how many samples are just in one test tube, that's just one piece of plastic. Right now I'm holding a test tube that's 50 milliliters, so about the size of like a mechanical pencil, I would say, and probably 1.5 times the width of a mechanical pencil, and it is packed to the brim with plastic pieces and small bits of plastic fibers. So that is how we process our samples. And you'll notice on the tube here we label them. So we have our sample site, Searim State Park. We have the date 9/14/19. HTL stands for high tide line, and then we have our transect that we did, so 50 by 1. That means we did 50 meters long by 1 meter. And then the T just stands for transect. So there's all sorts of little bits of plastic in there. So kind of think of it like a jar of marbles, except even smaller. So then what we do is we go through these and we take a picture of each piece of debris, catalog it, and then we enter it into an Excel spreadsheet with the measurements, the type of plastic. So was it a plastic particle? A nurdle is a spherical piece of plastic, and then if it was a fiber, and then we'll take that spreadsheet and do data analysis later. So I'll talk to you a little bit more about that here coming up.
The camera now shows
I'm going
Maureen's
to show
computer
you how
screen.
we take those photos from the dissecting microscope and actually get a more accurate measurement of each piece of marine debris that we collect. So this is done using an imaging program called Fiji, and it actually stands for Fiji is just Image J. So Image J was like the program before this one, so the people who made it were kind of being funny. So the name of the program is an acronym. But basically this program can do a variety of things, but what we're going to be using it for today is to get an exact measurement. So when you open up the Fiji program, it just comes up with a small ribbon under the main menu bar. So going from left to right on my MacBook, on the main menu bar, we have the Apple, we have the Fiji, where you can set your preferences, start and stop the program, File, Edit, Image, so File, Edit, Image, Process, Analysis, Plugins, Window, and Help. So the main things we're going to be using in the main Apple menu today are File and Analysis. And then if we go down to the ribbon for the Fiji program, we have a couple of different things like draw a lasso, draw a line, draw an angle, there's a paintbrush, there's a zoom in, zoom out, there's a little hand grip tool for navigating around images. But the main thing we're going to be using today in the ribbon bar on Fiji is this line tool. So to start, I'm going to go up to the File menu and I'm going to click Open. For the image we're going to be using today, I saved it to my desktop just to find it easier. But normally what we do is we take our images from each sample site and we give them all a certain name. And then usually the name contains the sample site, the date when it was taken, and some kind of a number to identify it. And then we store each of those in specific folders. But for today, I just put our image on the desktop. We click Open, and now the image that we took using the IPEVO document pen is showing underneath the ribbon for Fiji. Before I can do anything, I want to measure this piece of plastic, right? But right now, all this image is a series of pixels. It has no set measurement. So you'll remember before I mentioned a ruler, and what we can do is we can zoom in on this image using the Command plus button, and we can zoom in to where I can see the 6 and the 7 centimeters on our ruler in the image. I can click that line tool, and then I can take the cursor and drag it from the 6 to the 7 on the ruler in the picture. I'm going to go up here to the main menu. I'm going to click Analyze. I'm going to scroll down to our Set Scale option here. And now it's going to say, okay, the distance you have in pixels after you've drawn this line is 268. Then it's going to ask me for my known distance, which I'm going to put 1. We're going to keep our pixel aspect ratio to 1. And then it's going to ask me for units. So I'm going to put centimeters, because that's what our ruler is. Then there's something really important you need to check down here, and that means do you want to make your scale just for this line, or do you want to apply it to the whole picture? We say global. So this means we have 268 pixels per centimeter. We're going to say OK. Next, we're going to zoom back out so we can see our plastic. But
Turns out, because our scale is universal, we can zoom back in so we can see that plastic a little more clearly. We're going to use that line tool again, and I'm going to go from one side of the plastic to the other longwise. I'm going to go up here to Analyze and Measure. There's going to be a pop-up window that comes up in front of our photograph, and it's going to tell us the length was 0.572 centimeters. But just to make sure, I'm going to take a few more measurements here, both diagonally across the long side of the plastic, and for this I can use the keyboard command on the Mac called Command M, and it'll add another measurement to that pop-up window, and I'm going to do it again horizontally along the plastic. So we also got 0.521 centimeters and 0.480, so I'm going to take the highest of those three measurements. So our plastic in this photograph is 0.572. We would classify it as a plastic particle, and because this plastic was collected from Searim State Park in July, we would call it SRSPJUL, and then the color of our plastic in the screen is green, so that's what we would put it in the spreadsheet as Searim July, 0.572 centimeters, plastic particle, and green. And that is how we go through and measure and get an accurate measurement on our marine debris samples using a program called Fiji. So after we measure all of our plastics, we will put that data into a spreadsheet that looks something like this. So this spreadsheet has 4 columns and a lot of rows. So what I've done is I've taken our data from our 3 sample sites, Searim Beach, Galveston Island State Park Beach, and Mustang Island State Park, and put them all into one big long data set. So what we have here in our 4 columns is our first one is ID, and this tells us where the sample came from. So starting with Searim State Park, so this is abbreviated SRJUN for June. So this spreadsheet contains all of our data from June. Next, we have our material column, and our samples can be one of 3 materials. It can be either a plastic particle, a fiber, or what's called a nurdle. Next, we have our size, which was the actual measurement of the plastic, which is what we just went over doing in the program Fiji, and this is in centimeters. And then next we have our color, so blue, green, white, clear, brown. So in row 2, in our ID column, we have SR June. In our material column for row 2, we have it was a plastic particle. In our size column for row 2, we have it measured 0.243 centimeters, so this would make it a macroplastic. And then we have our color, which was blue. So this is kind of what our spreadsheet looks like for the month of June once we have processed all of those plastics, all of those marine debris samples, and processed them. Through how to measure our plastic particles and then how to put that into an Excel spreadsheet, I'm going to show you one of the programs we use to do some data analysis and draw some conclusions from all of this data. So the program I'm using right now, and that is on the computer screen, is called RStudio. RStudio is very common in a lot of biological, genomics, ecology fields. So the way RStudio works is you have 4 main, we're going to call them panels. So there's kind of in a grid. So in the upper left, we have our coding panel, which is the one that we type in. On the upper right, we have what's called our environment, and this just tells us like what we have coded or what we have made into what's called a variable, but I wouldn't worry about that for now. In the bottom right, we have our directory, so this just tells us where we're getting our data from. And then in the bottom left, this is what will give us our answers from the code we type in the active part of the screen on the top left. So the big things I want you to focus on are the top left and the bottom left side of the screen. Now, don't get too freaked out by all of the code. You don't need to know that. I just want to show you how we use this program to again, draw some conclusions from all of these samples that we have. So the first thing I had to do is I had to tell the program where to find that Excel spreadsheet. So I've set our directory over here to the folder where I have the spreadsheet saved, and I'm going to read the spreadsheet into the program, and I'm giving it a name, and I'm calling it Plastics June. So I've read the spreadsheet into the program, and then the way I know that it's read into the program is I go over to my environment and I can actually click Plastics June, and a new tab pops up in the upper left hand corner. And now, instead of that data being just Excel, you can see that it has been put into the RStudio program here. So now going back to my tab where I have my computer code, I can do something called a summary, and I can get a summary of our data from Plastics June. So for example, I can find out that for Galveston, which I have labeled GI for Galveston Island, we have 193 data pieces, or 193 pieces of marine debris. For Mustang Island, which I have abbreviated as Mustang Island State Park, we have 134 pieces of marine debris. And then for Sea Rim State Park, lastly, we have 154 pieces of marine debris. Another thing I can find out in the summary section is how many of pieces of debris were fiber, a nurdle, or a plastic particle. So fiber is anything like a rope or a stringy particle. A nurdle is a special kind of plastic because they are specifically spherical shaped. And then everything else is classified as plastic particles. So for fibers, we have 110 total. We have 22 nurdles total, and plastic particles, we have 349. I can also find out my minimum and maximum object sizes. So our minimum or smallest piece of marine debris was 0.57 centimeters. Our median or our middle piece of marine debris was 0.524 centimeters. Our mean or our average size of marine debris was 1.0 centimeters. And our max size of marine debris was 20 centimeters long. I can also figure out what color was the most prominent. So for example, we had 173 pieces that were blue, 112 pieces that were white, and so on. Now, say I wanted to find out a little bit more about each site specifically. Right now, the way I have the data in the spreadsheet, it's reading all of the data from all three sample sites. But say I just wanted to focus on one in particular. I can do this command called a subset, so it's basically splitting it up into 3 parts. So I can do that for all 3 sample sites, for Sea Rim, Galveston, and Mustang Island. And then I can use that same summary command for each of our sample sites. So say I do that for Sea Rim, I can get all those same demographics. So for example, we had 34 fibers at Sea Rim, 0 nurdles, and 120 plastic particles. Our smallest particle at Sea Rim was 0.057 centimeters. Our mean was 1.0 centimeters, and our max was 20 centimeters. And then again, I can also get the breakdown of the kinds of colors of plastics. So we had 76 blue pieces, we had 26 green pieces, and we had 6 orange pieces was our smallest. So this is just a quick way to get some quick information about the data that we have. Next, I just want to look at the plastic particles specifically. So again, I'm going to use that summary command, but first I'm going to do what's called a subset. So now I'm looking at plastics, but within all 3 sample sites instead of each sample site specifically. So if we look at plastics total, let's see here. If we look at just plastics instead of all marine debris, we have 145 pieces in Galveston, 120 in Sea Rim, and 84 in Mustang Island. And then if I want to figure out how many of those were microplastics and how many of those were macroplastics, I can do the subset and summary command again. So when it comes to microplastics, the sample site with the most microplastics was Galveston with 89 pieces of microplastic, and the site with the least amount of microplastics was Sea Rim State Park. And our most common color of microplastic was blue. When it comes to macroplastics, this is anything that's greater than 5 millimeters or half a centimeter. The site with the most macroplastics was C-rim at 62, and the site with the least macroplastics was C-rim at 17. And the most common color of macroplastics was white. So again, this is just to get some basic information. We can also do some plotting of our data in what's called a box and whisker plot. So for example, I can just look at the overall size of the debris at each of my sample sites with all three categories, plastic, fiber, and nurdle. So this just tells me that min maximum, which isn't really the most useful thing. So instead, I'm going to use this fancy code here to make a more detailed graph. And this graph will show us the difference in not only each of our three sample sites, our three beaches, but also our three types of debris, so fiber, nurdle, or plastic. So here we have a box and whisker plot with each of our beaches on the x-axis and then size on the y-axis in centimeters. And then we have three potential boxes in each beach, with red being the distribution of the sizes of fibers, green being the distribution of the sizes of nurdles, and blue being the distribution of the sizes of plastics. Now, one thing you'll notice is that most of what we see is red, because on average, we can see that fibers are bigger than everything else, because the red boxes are constantly above the blue and the green boxes at every single site. But what makes this graph not so useful is that you remember I mentioned we had that max outlier of like a 20 centimeter fiber. That's not really helping us visualize our plastic data very much. So instead, what I'm going to do is just visualize them separately. So going back to a different box plot, this is our three beaches, and this is looking at the size distribution of just microplastics. So you'll notice that for size on our y-axis, it only goes up to half a centimeter. And you'll notice they're all pretty much about the same. The box plots kind of overlap in a lot of areas at all three of our beaches. I can do the same thing with macroplastics, which remember all of these are going to have sizes over half a centimeter. And when we look at that, it looks like there's more variation, and the reason I say that is not all of these boxes are the same shape. So if we look at C-rim, it has a very small box between, it looks like 0.75 and like 1.2 centimeters. If we look at, that's Galveston. Then if we look at Mustang Island, it has a much wider range of like 0.75 and then 2 centimeters. So there's not as much overlap here, so we would just say that there's more variation. So this is just kind of a quick tool that we use to visualize some of our data. If you don't understand all of the graphs, that's perfectly fine. Just wanted to show you kind of what we do. So the reason I make all of these graphs is to get a quick sense of what does my data look like, kind of like sketching out ideas for a cartoon character or for a comic book. I'm still working on formal statistical analysis, so although my graphs, I can kind of infer that there's a difference, I won't know if it is statistically significant until I run specific mathematical tests, and that is my next step once we finish analyzing our July data from all of our microplastic sampling. I want to compare both June from all three beaches to July samples from all three beaches. So I hope you found some of this useful, and thanks for watching.